Genesis disperses enzyme substrate pairs to catalyze amino acids, perpetuating life. Amino acids are building blocks of proteins, while Genesis operates on a galactic scale. It emits biological photonic plasma that carries enzyme particulates. These particulates interact with interstellar dust, rich in organic molecules. Enzymes accelerate reactions, forming amino acids and seeding planets with precursors of life. It ensures that chemical conditions for life arise. You cannot force creation, but you can scatter so many seeds of possibility that life grows wary of choosing which ones to bloom. Plasmagenesis is genesis via plasma states, ionized torrents that cascade into coherent architectures. Stellar flares birth magnetic flux tubes. Plasma instabilities weave into current sheets. Magnetohydrodynamic turbulence crystallizes into filamentary networks. Magnetic reconnection events spawn plasmoids that propagate through space, carrying information encoded in their field generations. These plasma offspring inherit topological properties of their parent structures, creating genealogies of electromagnetic organization. Abiogenesis is genesis via non living matter, mineral substrates, and thermodynamic gradients organizing into replicating molecular systems through chemical processes. Hydrothermal vent chemistry spawns peptide networks. Clay mineral catalysis assembles nucleic acid polymers. Lipid vesicle formation creates first cellular boundaries. Meteor's heat at impact catalyzes amino symbiogenesis, creating life abiotically if conditions are arid and heat are high enough. Turbogenesis is genesis via turbulence, chaotic flows that organize into stable systems. Dust devils form stars, turbulent flow sorts biology, reaction diffusion patterns are created. Cardiovascular turbulence sorts blood cells by size and deformability, creating laminar core flows surrounded by cell-free plasma layers. Turbulent mixing in embryonic development generates morphogen gradients that establish body axis patterning. Chaotic advection ensuring proper chemical signal distribution. Symbiogenesis is when outer space aminos combine with terrestrial aminos to form proteins. Mitochondrial work creates eukaryotic powerhouses. Bacterial partnerships weave into multicellularity. Viral integration rewrites host genomes into hybrid architectures. Separate evolutionary trajectories converge into unified organisms. Mismatched amino acid stereochemistry prevents proper protein folding, causing extraterrestrial and terrestrial sequences to aggregate into insoluble tangles. Incompatible codon usage between cosmic and indigenous genetic systems creates translational errors that produce truncated, malformed proteins. Cryogenesis is origin through freezing, preservation that leads to eventual awakening. Time stop matter becomes Genesis seed in future ages. Halting cellular metabolism, ice crystal formation creates protective lattices around molecular structures. Vitrification prevents freezing damage through glass transition states. Cryptobiosis demonstrates reversible death through desiccation and freezing. Cellular architectures reorganizing into crystalline arrays to preserve genetic information across geological timescales. Permafrost bacteria remains viable for 20 million years, metabolic machinery locked in ice but ready to resume function upon thawing. Cryoprotectant synthesis allows organisms to survive temperatures that would normally shatter cellular membranes. Pyrogenesis is genesis through fire. Catastrophe is creation. Combustion births ecosystems and stars. Thermodynamics increases entropy to create quantum complexity, culminating in life. Wildfire clearing forest undergrowth triggers seed germination. Volcanic eruptions create new landmasses from molten rock. Ecosystems depend on periodic fire cycles, their reproductive strategies adapted to pyrolytic triggers that release nutrients from accumulated organic matter. Serotinous cones open only under extreme heat, their seeds dispersing into ashen-rich soils where competition has been eliminated. Electrogenesis is formation through electric field dynamics, sparks that trigger complexity. Lightning initiated reactions, nerve conduction, synaptic ignition. Lightning strikes catalyze atmospheric chemistry. Action potentials propagate information through neural networks. Electrochemical gradients drive adenosine triphosphate synthesis in cellular powerhouses. Electric fields become organizing principle that transforms random molecular motion into coherent biological function. Radiogenesis is formation through exposure to ionizing radiation, mutation hotspots, radiotrophic life, stellar core evolution. Cosmic ray bombardment triggers atmospheric chemistry cascades. Gamma radiation induces crystal lattice effects that create new material properties. Neutron activation transmutes stable elements into radioactive isotopes that decay into entirely different atomic species. Radiotrophic organisms harness ionizing radiation, their melanin-rich cell walls capturing gamma rays, then converting them into metabolic fuel through radiolysis of element 1 to the 2-8 molecules. These extremophiles thrive in reactor cores and contaminated zones. Mutagenesis is replication errors, tautomeric shifts, spontaneous deamination, deoxyribonucleic acid slippage. Mutation hotspots cluster around deoxyribonucleic acid damage sites where radiation-induced double-strand breaks trigger error-prone repair mechanisms, creating genetic diversity through controlled genomic instability. Retrogenesis is when retroviral integration rewrites host genomes by inserting ancient sequences into modern chromosomes, awakening dormant evolutionary programs. Transposable elements jump backward through phylogenetic time, carrying genetic memories from extinct lineages into contemporary organisms. Carcinogenesis is genesis via cellular rebellion. Normal growth controls abandoned to spawn immortal cell lineages that rewrite tissue architecture. Oncogenic transformation liberates cells from contact inhibition, metastatic invasion creating new ecological niches, tumor angiogenesis hijacking vascular development to serve malignant expansion. Cancer becomes evolution accelerated beyond organismal constraints. Porogenesis is genesis via pore formation, void spaces organizing into interconnected networks that enable transport, storage, and emergent functionality through controlled emptiness. Vesicle trafficking creating cellular compartmentalization. Cars cave systems channel groundwater flow. Nanoporous materials exhibit molecular sieves.